The goal of this video is to cover cell membrane structure and function in a more thorough way. So if you go back in the playlist a little bit further back, we talk about the basics of lipids and phospholipids and how they form membranes. But now we're going to get into the details of membrane function and transport across membranes through channels and active transport pumps. So as a quick refresher, a membrane is actually a mixture of a few different things. The main component that's absolutely critical for cell membrane function are phospholipids. So all these molecules you see here that have heads pointing outward and tails pointing inward, those are your phospholipids, and they're the molecules that spontaneously form membranes as soon as you mix them together. More on that in a minute. Within the membrane, you also have cholesterol molecules which stabilize the membrane and control its fluidity, right? How liquid versus solid it tends to behave. And then on top of that, we have proteins that are embedded in the membrane, right? Integral protein really just means that it's integrated or incorporated into the membrane, and those proteins can act as pumps or channels like transporters. They can also be modified with carbohydrates that turn them into what we would call a glycoprotein that help identify the cell to the immune system. And lastly, we also have connections between the cytoskeletal proteins. These yellow fibers on the inside are representing protein fibers inside the cell, and these purple guys on the outside are representing extracellular matrix proteins. So our membrane also is trying to link intra and extracellular proteins to stabilize the positioning and structure of cells. Right, so phospholipids. Again, more details on this in a previous video if you go back a few chapters in the playlist. But as a recap, the idea is these hydrophobic tails are made from fatty acids. So a phospholipid is really just two fatty acid tails connected to a glycerol in the middle that links them together. And then on the very top, you see a phosphate molecule, this PO4-. And the point of this is the fatty acid end is supposed to be very hydrophobic and not like water, and the phosphate head is supposed to be hydrophilic, meaning it does like water. And so when you add phospholipids into a water-based solution, they very spontaneously form membrane structures, right? Because the tails like to get away from water and bury themselves on the inside of the membrane, and the phosphate heads on both sides like to interact with water because the intracellular environment and the extracellular environment on both sides is going to be water-based. Right, so if you take pure phospholipids, add them in a liquid solution, shake them up, this is what you naturally get, a lipid bilayer that tends to form a sphere. And the reason for that is just that it's the most energetically favorable way that this can happen because everyone's happy. All of these phosphates on the outside and the phosphates on the inside get to be exposed to water, right? Because you'll have water in the middle, right? That is generally the most common way that this happens. You can also get single layered spheres if the fatty acid tails entirely interact with each other. But since cells are large enough to have a hollow space in the middle, we end up getting something that looks more like this. And if you zoom in on any one particular portion of the cell membrane, right, it should look like this. Lipid bilayer, phosphates out, tails in, and then cells can start deciding how many of those integral membrane proteins they want to insert into the membrane just based on their function. Right. When that happens, these integral membrane proteins, they have to actually span the cell membrane. And in this picture, the yellowish, right, off-white rectangle, that is supposed to represent the actual membrane itself. They're just not drawing in the lipids. And then these structures in pink and blue are supposed to represent proteins. So the point that they're trying to get across is you can have different numbers of passes through the membrane. So this single pass protein really just means that it has a single chain of amino acids moving through the membrane. And what you would expect to see is the amino acids that are inserted in the membrane are going to tend to be nonpolar, which, remember, nonpolar means the same thing as hydrophobic, right? So those would be hydrophobic amino acids. The parts of the protein that are intra or extracellular would tend to be hydrophilic, right? Because they're going to interact with water. So in a protein like this, right, this is a 3-pass or 3 transmembrane pass protein. You would expect these to be hydrophilic, and then every pink region would be hydrophobic. So if you want to insert a protein in a membrane, all you have to do is alternate hydrophilic, hydrophobic, hydrophilic, hydrophobic over and over again. That will give you your multiple passes. And sometimes it can be a very large number of passes. And here, each of these blue rectangles represents one span through the membrane, and they wrap around in a cylinder. So this is what you would use to form like a facilitated diffusion channel, right? That's going to let something pass through the membrane, like sodium ions or glucose. We'll see examples of this in just a bit. Another type of protein molecule that you commonly find on cell membranes is receptors that regulate how your cell interacts with other cells and to detect things that need to be taken into the cell by endocytosis. So in this example, this cell that we're looking at would be a white blood cell, specifically a CD4 positive helper T cell. And those happen to be the cells that HIV infects. And so when the actual virus enters a person's body, it can't replicate on its own, right? HIV needs to get inside of those target cells because viruses aren't really alive, and so they have to trick their way in, right? CD4 is just a integral membrane protein that's found on those T cells, and HIV has evolved to bind the CD4 receptor as a way to trick the cell into bringing it in by endocytosis. And so CD4 is supposed to recognize normal human molecules, and when that happens, bring them into the cell through endocytosis, which is something we'll mention later in more detail. HIV has just sort of gamed the system by evolving to bind the receptor. That's how it sneaks its way in. All right. This next concept, membrane asymmetry, we've already seen this. We're just pointing out why it matters. Asymmetry really just means that the two different sides of the membrane, the exterior as opposed to the interior, are going to look a little bit different, right? Some of these proteins span the entire membrane. Others are peripheral membrane proteins that are only found on one side or the other, right? Like these guys. And that makes sense because the proteins on the interior have to bind to these cytoskeletal proteins, and the ones on the exterior have to bind to extracellular matrix proteins. And so you need the inside and the outside to have a slightly different composition because they have a slightly different function. Okay, so the next place that we're moving on is the concept of diffusion. And the first question that you always have to ask is, can your molecule or ion diffuse directly through a cell membrane? So if it's possible, right, if whatever this molecule happens to be is able to diffuse straight through the membrane, you're always going to get movement, net movement, from high to low concentration, right? And by concentration, you would just be measuring this as milligrams per liter or millimolar or something to represent how much of that molecule there is. And if you track what would happen over time, you would start getting diffusion in this direction until at some point, like over here, you would get to the point where the concentrations are the same on both sides of the membrane. Once you've reached that point, right, we say that we're at equilibrium. And equilibrium doesn't mean that it stops diffusing. It just means that you're going both directions at the same rate. And so the amount of molecules on both sides doesn't change. They're just trading places with each other at equal rates, right? It would kind of be like if you're in a classroom and every once in a while someone gets up to leave, but as soon as they do, another person walks in from the hallway. You would have movement through the door, but the number of people in the room wouldn't really be changing, right? Now, in order for this to happen, the only molecules that can diffuse directly across a membrane, we call this simple diffusion. That only happens if the molecule fits a few descriptors. First off, it has to be relatively small, and it has to be nonpolar. And remember, nonpolar means the same thing as hydrophobic. It just means that it likes fats, so that when it wiggles its way through these phospholipids, it doesn't get just rejected and kicked back out, right? If it's soluble in that lipid layer, it has a chance to move the whole way through. So molecules that are too large, or if they're polar or have a charge, they will not get through, right? And most molecules don't, right? Diffusion directly through the membrane is relatively rare. All right. Now, assuming that your molecule can move through the membrane, that's what would happen. It just starts diffusing from high to low until you hit equilibrium. Now, if your molecule cannot move through the membrane, then you can get something called osmosis or osmotic pressure. So in this example, we've got our beaker set up with a semi-permeable membrane, and in their example here, these purple dots, whatever solute that is, pretend that it's, I think they say sucrose, just table sugar. If that sucrose can't get across the membrane, but water can, what will end up happening is, instead of the sucrose molecules diffusing, water will diffuse. And so osmosis is just the diffusion of water. And the thing to always remember is water follows salts or solutes. And so if the solute concentration is higher on this side, water is going to go that way and keep diffusing until now you see the concentrations are very similar, right? They're representing that by the molecules being equally spread out on both sides. Right? And that water really, very literally did move from one side of the beaker to the other. It might not be this extreme, but you can look and actually see a difference in the water level. That is osmotic pressure, the pushing of water across the membrane to try to reach equilibrium. Right. This concept matters an awful lot for actual living cells because if you have diffusion gradients and water moves, you can make cells either expand and burst or shrink and shrivel up. And so tonicity is really just asking the question, is the concentration of solutes, remember solutes just means dissolved molecules, is it the same on both sides of the cell? If it is the same on both sides, we say that the cell is isotonic, meaning the concentration of solutes inside the cell and the concentration of solutes outside the cell is equal. Right? Then movement of water in and movement of water out is going to happen at the exact same rate, and the cell stays its normal size. That's what we want. Right? Hypotonic means that you have a relatively low concentration of solutes in your solution. Right? So if the cell is in a hypotonic solution, it just means lower solute concentration outside, and so by comparison, you would have a higher solute concentration inside. The effect of that is, remember, water follows solutes or salts. And so the water will rush into this cell and make the membrane expand, and eventually it can burst. And so when we say the cells are lysed, that just means the membrane's ruptured and it popped like a balloon. If you're hypertonic instead, that means that you have a relatively high solute concentration on the outside of the cell. So you would have a relatively low concentration inside. Then water would rush out, again, always moving towards the area of higher concentration for water, and that would make the cell shrivel up, right? It might not burst as easily, but they're both pretty bad as far as outcome goes. So the conclusion from this is, like in a hospital, when someone gets a fluid infusion, like from an IV bag, you want to give them isotonic saline, right? They'll add some sodium chloride to the fluid to make sure that it's isotonic with the cells, right? Because if you added pure water, you would wind up in this solution. Hypotonic, cells would burst. If you gave them fluid that was too salty, you would wind up in this situation. Cells would shrivel. Right. Okay, so now let's talk about transporting things that we want to get across cell membranes. Right. So in your list of things that can and can't move across cell membranes, remember, only molecules that are small and hydrophobic or nonpolar can make it across membranes. The vast majority of molecules in a cell do not fit either of those characteristics. So examples that are very common, gases like CO2 and oxygen, they can very easily diffuse directly across membranes. They're small, they're fairly nonpolar. That's how your lungs can exchange gases without having a transporter. For anything else, for ions like sodium, anything with a charge generally can't be moved across the membrane. So sodium, potassium, chloride, calcium, those are all hard no's. Those molecules do not move through membranes, they'll bounce right off. Same goes for any carbohydrates like glucose, most amino acids. These molecules, they can't diffuse directly across the membrane, so they're going to need a transporter. All right, so this next handful of slides, we're going to be going through different categories of transporters and their functions, and you can follow along to that to the next video. I'm going to stop it here, but that'll be the next one in the playlist, so make sure flag the playlist and subscribe so that you get all these videos in context.